What was Paige saying? She's talking.
I don't know. I think she's talking to herself out loud.
That's fine. They say smart people do that. I think that's to make them feel better, though. All right, here we go. Finally, hallmarks. Okay, so the next one we do is enabling replicative immortality. You've learned all this information. This is us reviewing it just in a different way. So we learned in one of our chapters that there is the Hayflick limit. So our cells in our body, we have stem cells, and then we have the cells that are doing the work. So I'm just going to call them work cells. Meaning, like for stem cells, if you think in your... Oh, this doesn't show up very well. I'm going to turn off the other set of lights. So, oh, nighttime. All right. So stem cells. Let's think of bone marrow. In our bone marrow, a whole bunch of multipluripotent stem cells that will eventually grow up to either be white blood cells or red blood cells, right? So stem cells replicate. Work cells usually, don't. They've hit their final form. So eventually, some of these stem cells will have replicated enough to where the ends of the chromosomes shorten each time you go through a replication, and then you've lost the end of that chromosome. So when that happens, we say the cell can no longer divide. It causes itself to go through apoptosis because these ends are not protected anymore. They need to be protected. Several splits or several divisions of these cells will cause the ends to get shorter and shorter. So the number of divisions that you can do, that's called the Hayflick limit. There's a pretty cool article at Arizona State about Louis Hayflick. That's the guy who discovered it. So what happened, there's a guy before him that said he had cells in culture for like 50 years. And he was telling everyone, it's not about the cells, it's about what you put on the cells, the nutrients to make the cells keep going. So everyone at that point, before Hayflick came on the scene, thought that cells could just live forever and ever, forever, ever, as long as they had the right nutrients. And then Louis Hayflick came on the scene and said, no, that's not happening. Whatever you're seeing is not true. Big controversies between the two, but finally Hayflick was deemed correct that over time cells will hit their limit because the telomeres are too short. So the longest you can see are like 50 cycles. Those are fibroblasts. Fibroblasts are probably the hardiest cells in our body. Fibroblasts are part of cartilage, kind of like extracellular matrix, holding things together all across our body, everywhere. When you make cells, they're the easiest ones to make. So after this these 50 cycles, the cell will enter what's known as senescence. We've talked about it before, and that's cell arrest, and it should lead to apoptosis. And that whole process is normal and it's protective. What else do I know about Hayflick? Hayflick is still alive. He was born in 1928. And I'm just imagining he's close to dying, but will he? What if he figured out how to extend his telomeres? And he's not telling anyone. And then he's just going to say, I just kept eating and I had the right nutrients and cells can live on forever and ever. He's a liar. Hayflick's a liar. He also got in a giant fight with the NIH and sued the NIH and eventually actually got the rights to the cells that he made.
As he should.
Well, good, interesting question. Should he? Because the cells he was working on were infant heart cells.
Like babies.
Baby.
Oh no. Oh no. How did he get those?
Right. How did he get those? Should he have had control of them or should the parents?
Wait, well, did the parents give the baby cells? Like, how did... Like, did he just say, hey,
Hey!
take this baby cells, yeah.
I don't know. Back in the 60s, that ethics was all loosey-goosey. If you think in the
I
50s,
almost just said I didn't think he was alive in the 60s, but he was. So.
yeah, time, good. So there's just a lot of controversy that surrounds him, but everything that he's done so far has proven to be true, and he won his cases, and yeah, and those cells that he made are still growing. They were, I think, helped find the vaccine for rubella, against rubella. All right, I think that's all I can remember about Hayflick. Very cool guy. This won't advance. Why won't it advance? There we go. All right, senescence. So again, basically growth arrest. Many people will say it's irreversible, but that is what cancer cells can do. They can put themselves in senescence, and some other signal can wake them up. So I don't like when some sites or some scientists say irreversible, because it can be, but it's just not normal. A healthy cell that went into senescence would not come back out of it. It would go to apoptosis. So what regulates senescence? All our tumor suppressors, P21, which we haven't talked about, but P53 does, and RB, we know what those two do. P21 is just another type of tumor suppressor. So if there's something wrong with those tumor suppressors, then the cell may not go through senescence, but keep replicating, and those telomeres, the ends of those chromosomes are gone, and that's going to just lead to more issues with chromosomes. So what can lead to it? Well, repeated cell culture. So let's talk about what that means. We've got primary cell lines and then transformed cell lines. Okay. Primary cell lines are cell lines you took directly from the body, someone's body, whether it be a non-human mammal or a human, and these are the cell lines that have not had any mutations, and so they will hit, you will see them hit their Hayflick limit. For instance, like liver cells. You can take a liver, dice it up into tiny little pieces, add some collagen and some media nutrients, and they will grow. You can split them several times, but I think after like their 8th split of liver cells, they die out. So they hit their Hayflick limit somewhere within a month of having created them. That's considered a primary cell line. Then you have transformed cell lines, which would have first come from some type of organ, but some type of mutation has caused them to become immortal. So again, this is either someone in the lab caused them to become immortal, or they spontaneously became immortal. Okay, so, and I'll talk a little bit more on that in the next slide. Other things that will cause senescence, obviously we just talked about the telomeres getting shorter, radiation, if it's an oncogene activation that wants to deal with telomeres, then that might cause it. And then big thing is oxidative damage, because that's really done by the human. What type of diet you have, healthy lifestyle, other environmental factors you might come in contact with, and that's oxidative damage that will cause the cell to go into senescence. But senescence isn't all that bad. It can do some good things, and mainly when a fetus is growing, because you can imagine that there's times where you need to cause apoptosis. So I think we talked about this, that your hands are first web hands, so your hands will look like a duck in the beginning when you're in utero, and then senescence will occur, and then apoptosis to basically take care of any of that extra skin that would have been between our fingers, so we don't come out like ducks. So senescence, good for that. Wound healing, we have actively going on in our bodies. Senescence helps with that. And then trying to push something that might be going into a tumor phase into just senescence and killing it. So senescence is not all that bad. It does play a normal role. Ooh, okay. So here are all the different things that could cause senescence. So things I want to focus, let's just look right here, At the center of this pathway, many of these pathways, is P53. So basically, it's saying, let's look at this, excessive calorie intake. So that's the top reason, the one reason why we might lead to senescence. Because excessive calorie intake can lead to obesity. Did I just spell that wrong? I did. Okay, obesity, and then it will lead to inflammation. And anytime you think about inflammation, think about reactive oxygen species, right? This is a pathway we've already talked about. And then it has, you can see it's going to interact with the RAS-RAF pathway. We kind of talked about AKT. We've seen mTOR, all our little proteins. But eventually, what should happen is P53 will realize that something bad is happening and then should shut down the CDKs and the cyclins. Because remember, cyclin-dependent kinases and the cyclins come together to push you through the cell cycle. Well, now P53 is going to stop CDKs and cyclins from pushing you through the cell cycle. So this would be an example of something you could put on your presentation. Here's the pathway that can lead to senescence. And this is a good pathway that naturally occurs in our body when something's wrong. So again, if we get radiation, lots of carcinogens, heat shock, or any type of metabolic issue, will get released. And then, of course, we have cytokines will get released, which turns into a bad thing because it'll just keep activating reactive oxygen species. But that's just part of cancer. If we keep the area inflamed, then we continue to build reactive oxygen species. So just have a healthy lifestyle. Oops. Okay. The paper talks more about telomeres, but all of that has to do with leading to senescence. So at some point in the paper it said, when telomeres get short and we stop, that will stop cancers. And then another part of the paper said, well, sometimes that starts cancer. It's actually all talking about senescence. When are we promoting senescence and when are we causing it to stop? So what I want to show here is that senescence, depending on what's going on in the cell, can either stop tumorigenesis or it can promote tumorigenesis. That just tells you that our cells are very complex and there's lots of things going on. So sometimes senescence is great, it stops the tumors. Sometimes senescence works against us and it promotes tumors. Same thing with telomeres, stops it and sometimes promotes it. Okay, so why do we care about those telomeres? So remember, if this is just the chromosome, the ends have these repeats on them. And then after you go through cell division, your telomere will get shorter and the repeats will get shorter until the end where there are no repeats there. So the telomeres are gone. So what can happen? In this situation we're seeing these ends have come off, the telomeres have shortened, and now we see sister chromatids being linked. So before, those were two separate chromosomes. Let's say they're both chromosome 8 because they were sister chromosomes, but they fused together at the top. So when you take away the telomeres at the end, these become sticky ends. Right, so you can see how bad it would be to have two chromosomes stick together. Same thing, we've actually seen this already happen. The non-homologous end joining, that's NHEJ. I'll spell it, but I'll spell it wrong. Non-homologous end joining. Maybe spelled it right. So here we have two different chromosomes. I want you to think about them as two different chromosome sets. So let's say this is chromosome set 8 and this is chromosome set 10. Once we take off the telomeres, now you can see for this middle one, chromosome 8 has joined chromosome 10. So you have 8 and 10 now stuck together. Again, anytime you stick two chromosomes together, that's bad. They should never be stuck together. They should be able to come apart. Okay, so those are things that can happen when telomeres get too short and the cell hasn't gone into senescence. If there's no senescence, then we're going to cause these funky alterations to occur, which leads to more mutations. So, if no senescence, telomeres get shorter, they turn into very sticky ends, and then we end up joining different chromosomes together, causing more mutations. All right, so how do you become immortal? You can't. Well, I'm sure in the future they'll figure it out, but right now we can't. But, so how is it that the HeLa cells that we work with in the lab, so the Henrietta Lacks cells, how is it that they became immortal? So I'm going to write that up there. HeLa cells were spontaneously immortal. Okay, so they, immortal have two M's here. There's a cancer in the cervix, and it was there for so long. I mean, that's what cancer cells do. They become immortal. Now, outside, let's say I want to take your liver cells, and we talked about how if we keep splitting your liver cells over time, they will die out. That primary cell line only has a limited Hayflick limit, and they die out. But I really, really wanted to work on those cancer cells. So I can do three things. I can keep splitting them and hope that a spontaneous mutation takes place. Okay, but that could be 10 years of me splitting the liver cells. Of course, we know they're going to die out, so it's not really going to work. Spontaneous mutations are difficult to wait for. Or we can insert this protein called TERT. It's basically a telomerase that will become active and help rebuild the telomeres. So we are taking a cell. So I got one cell, I don't care where it's from, and we are injecting it with TERT, so that rebuilds telomerase activity. So if we keep telomerase activity up, then we can cause it to be immortal. The telomeres, or the telomerase, will keep building the ends on those telomeres so that they never shorten and never become sticky. Okay, so that's one way we can do it. The other way, we've already learned that viruses can get into our cells, and we can use viruses, this simian virus 40. It has this thing called T antigen. For some reason, I actually don't remember the science behind it, but if you put SV40 into your cells that are in culture in the lab, and let them grow up after, I think, a couple of splits, eventually that T antigen will cause the cells to become immortal. So these are two of the most common ways. If I want to work with a specific cell line, somebody has a very unique cancer that we haven't seen or we haven't done a lot of research on, and you want to make sure those cells keep growing in culture so you can continue to study them, you might insert the TERT proteins or you might insert the SV40 T antigen virus into those cells. All right, so there's two downsides to these two methods. You're putting something into a cell that wasn't naturally there. And granted, this might be the only way we can study that type of cell line. You've changed that cell line from what it normally is. So every experiment you run on cells that you made immortal by either TERT protein or SV40, you have to take with a grain of salt because you've changed them in the lab. We assume, we're crossing our fingers that it works the same in humans, but we can't say that with 100% certainty until you start testing it with other human cell lines. Okay, so immortal cells, great for working with in labs, but do change up the inner workings of the cell. And then here, just like with senescence, the book or the paper did talk about how telomerase, we know it leads to aging. We know that if telomerase is no longer working, it can also lead to tumorigenesis. But it does have healthy roles in just normal structure of the DNA. And then it can also help fight off DNA damage or tell the cell to go through apoptosis. So if if I can make this image more messy, this side is useful for us because, well, we all age. We're going to die at some point. But we all do gene expression for certain proteins. And all our cells should end up going through apoptosis after it's past its stem cell life. But what we don't want is that tumorigenesis. So there's a lot of research on that telomerase. I would say there's more research on telomerase not because of tumorigenesis, but because of aging. People hate aging, and as your telomerase decreases in activity, that's what causes aging. All right. Are there any questions on telomerase? All right. Inducing angiogenesis. Another word for angiogenesis is neovascularization, just in case you read it other places. So the idea is a cancer cell wants more blood vessels towards it. This is what a normal blood vessel would look like. And then if a tumor is tricky enough, it can cause a blood vessel to come towards it so that it gets blood supply. So that's what we call tumorigenesis, is the making of a new blood vessel. So let's get into how it affects the cell. Oh wait, these are the different versions of angiogenesis. All right, so what are we looking at? This top one, hold on, my notes are gone. Okay. This top one is called sprouting. So something is telling it to start growing that way. Okay, that's normal. So maybe you are working out a lot and you've started to build more muscle. Those muscles need more blood vessels. And so the muscles will release a growth factor and it will kind of call the blood vessel over it to the new muscle. So you kind of get this sprouting to happen. There's another way where we just split it. So this was one vessel and blood was just flowing through. And then we decided to, maybe we can divide this blood vessel into two. So this will eventually divide into two separate blood vessels. Okay, so that's the splitting. Then there is this vasculogenesis. So this is more when we see in neonates, and in embryos. This is just the making of the blood vessels. So in the beginning, you're just a bunch of cells, right? You're just this ball of cells, and slowly they start to change. And we call these EPCs, or endothelial, or endothelium, progenitor cells? Progenitor cells. Okay, so they're just telling us these are basically the stem cells that happen in an embryo that start the formation of blood vessels. So this is 100% completely normal, needs to happen as the embryo starts to grow and organs start to form. You got to get blood vessels to it. You also remember we just talked about bone marrow, that has a lot of pluripotent stem cells. So here's just another, now you're more of a grown being, but you're going to cause the formation of more blood cells. So we got, again, those recruitment of those endothelial progenitor cells. If you don't remember what endothelium is, that is just the cells that make the blood vessel. So that's a cell, and we link it to that endothelial cell, and we link it, well, now it's going to become an endothelial cell, but it's the progenitor, and then that's an endothelial cell, and that's an endothelial cell. That's an endothelial cell, so you get the picture. That's what makes the tube, our endothelial cells. On top of the endothelial cells, you have to have something called pericytes. Pericytes are almost all over the place, but we think of them more with endothelial cell because they are the fibrous connective tissue that basically hold the blood vessel in place so that it remains a tube, but also stays where it should. So we'll talk about pericytes later. All right, so this is neat. These are tumor cells. All of these gray things are tumor cells, and they are forming a blood vessel, but they're not really a blood vessel. So they're just making their own tube and hoping that blood will lead to the mass tumor. That to me is pretty ingenious by tumor cells. Go, let's just try to create a blood vessel. Let's just all link together and hope that we can make it, the blood vessel's leaky and we can just have the blood flow towards the tumor. And then, as we all know, cancer cells can turn into other cells, so now they've turned themselves into actual endothelial cells. So they've gone through a mutation and changed. Okay, so those are the different ways we can form blood vessels. All right, so I told you that we would talk a little bit about the microtumor environment. The reason we're talking about this is to understand why a blood vessel would start to grow. All right. In the blue is the new tumor. Don't think that the blood vessels are there yet. They're not. So whatever you see in red, those blood vessels aren't there. So there's just this mass of cells that have fibroblasts around them. We just talked about fibroblasts. They keep things together. They're all over our body, but they're specific cancer-associated fibroblasts that keep them as a ball. There's also, you've got some white blood cells and some other, these are still types of immune cells. So any tumor should bring in natural killer cells, because we know natural killer cells love to fight off tumors, but it's also going to bring in other immune cells, and those immune cells think that they're attacking the tumor. The tumor's so tricky that it normally tells the immune cells, well, it either can hide from it or it tells it to do something different that helps the tumor. Same thing with some of these other types of immune cells. So dendritic cells are the immune cells that are in our skin. Macrophages are all throughout our body. Okay, and then other things that you might see, because there's so many immune cells, are different chemicals. So cytokines and chemokines, these are just signals. And again, if the tumor is super tricky, it'll figure out how to turn these signals into helping it grow. Okay, so all of that, the fibroblasts, and we'll see periocytes later, and all the macrophages, and all the other white blood cells, and all the signals that come from it, that is the tumor microenvironment. It's the tumor along with all the other cells associated with it. So what does that have to happen with angiogenesis, and how does it start? Because of this. Okay, again, I want you to focus on the blue cells, not the blood vessels yet. Inside this really densely packed tumor are the stem cells of the cancer tumor. So in the middle are the stem cells. This is the heart of it all. But it's so dense in there that those tumor cells are never getting oxygen. So they're deprived, they're in a hypoxic environment, and they will tell the nearby outer tumor cells, can you please release some signals to draw some blood vessels towards the center so that I can breathe, essentially. So the cancer cells that are in the middle can breathe. That's really the beginning of the signaling for angiogenesis, is the fact that it's created a hypoxic environment in the middle of the cancer. So it's not really, the cancer cells don't purposely make it hypoxic, it's just the situation of the cells that in the middle there's no oxygen being delivered. Everything else on the outside of this picture are showing you other types of things that you would find in the tumor microenvironment. So again, we talked about periocytes, which help hold blood vessels together. Here's a periocyte right there. See, it's on the outside of the blood vessel. We talked about fibroblasts holding everything together. We got macrophages, we got more immune cells, platelets, and then those endothelial progenitor cells, endothelial cells making all that up. So that's what the tumor microenvironment is, and then inside the tumor is that hypoxic environment. All that will eventually lead to the signaling. Okay, the signaling here, we will call this the tumor cell. And we will call this the blood vessel, which it says right there. So in the middle, you can see that it's created this hypoxic environment. It says it's in hypoxia. And there's a system, this is, HIF is a hypoxic initiation factor. It helps start the release of these signals to call in a blood vessel. So this is just a protein, transcription factors. They're made when it's low oxygen environment. They will go and bind to certain genes that will promote the release of growth factors. Okay, so now these growth factors get released. VEGF is one of the most popular ones, vascular endothelial growth factor, right? So those will bind into wherever it needs to bind, and then eventually you get more migration of the blood vessel, vascular permeability, things like that, things that will call blood vessels up towards the tumor. Okay, so the main issue is it's a hypoxic middle. So these hypoxic factors will cause the expression of genes that will release growth factors that will interact with the blood vessels to call it towards the tumor. Okay. So this is what we're seeing as those growth factors are released. So up top is a normal blood vessel. Down here we have a small little tumor, but in the center, those darker blue cells are in a hypoxic environment. So they are releasing all these different hormones. These are all hormones, VEGF, EGF, so vascular endothelial growth factor, endothelial growth factor, fibroblast growth factor, TGF-beta, which is tumor forget, and then insulin-like growth factor. You don't have to memorize all of them. Again, all signals that will go up, and look, it's going to interact with the blood vessel. You see EGF, VEGF, all interacting with the blood vessel. All right, so that's the start of it. But look, it also calls in those MMPs, I can't remember, what is it called? We'll look it up later. But anyhow, these are the ones that help with the transition through different levels of tissue. If it's going to be metastatic, it helps, the MMPs are released to help the tumor move through the tissue and get to the bloodstream. But how did those MMPs first get there? That's because the tumor was releasing it to call the blood vessel down. So those MMPs will chomp their way through, allowing that blood vessel to kind of break through. So the MMPs are first there, not to help it move, but to help bring the blood vessels in. It's just great that the MMPs stay there for the cancer so that it can eventually move. All right, well, great in a bad sense. All right, we got the sprouting that we saw, those little tip cells that will cause it to sprout. Then here you can see the stalk and it's growing. This tumor's releasing more VEGF. And then finally you see that we've got a blood vessel there, and we've got some other things that are going to try to hold the blood vessel together so it can deliver nutrients. All right, so those are the common steps. But let's look on how awesome or how non-awesome our tumor cells are at making blood vessels. Okay, up top is a normal structure of a blood vessel, right? Endothelial cell, endothelial cell, endothelial cell, making the tunnel. And that's normal, everything's great. Cancer cells, one, we saw how they can make fake blood vessels, but two, even when they get the chance and a blood vessel is coming towards them because they've released all their VEGF signals, they make really lousy blood vessels anyhow. This is a blood vessel. This is a legit blood vessel, but look how it's arranged. It's leaky, it's thicker, it's not a good blood vessel. So they don't even make proper blood vessels, and here's how you can see that's the lining of blood vessels. That's a normal picture. And then the lining of blood vessels made by tumors, again, are very thick all over the place and, again, leaky. So it's not, the tumors don't even do that great a job. But to be honest, having it leaky helps the tumor, because instead of it creating this straight line right into the middle of the tumor, it now creates this leaky, so nutrients are getting all over the place and just helping that tumor grow more and more. It does a bad job, but it helps it. All right, and I think, is this our last slide? Yeah. So since I asked everyone to talk about a drug that interacts with one of the hallmarks, this is a slide showing you some drugs. I think if I, hold on, discard. All right, so you can see that there are a ton of drugs out there that work on angiogenesis. So if you wanted to, you could just look at this and go, oh, I'm going to tell everyone what, I can't even say that, this cetuximab, what that drug does. So we know that it goes after the epidermal growth factor receptor, and if we can shut down angiogenesis, then it will not cause growth of hormones or growth of blood vessels, and this works very well in squamous cancer of the head and neck and colorectal cancer. So if you were going to create some type of pathway example, you would first start up with this, right? Because we said hypoxic environments will cause the activation of genes that will cause the release of growth factors and recruit in blood vessels. And then how do we stop that? Well, we can pick one of these monoclonal antibodies and stop the signaling or stop the receptor from filling up with growth factors. Oh, that makes sense. Okay, that's it. We did it. 4 and 5.